Chapter 3, the cell. We'll try to get through this quickly, roughly 40 slides. I'll give you guys the highlights. Cells, we've been over this previously. We know that cells are the basic unit of life. All the biomolecules and biochemistry we have discussed need organized. So we're going to now talk about the prokaryotic and eukaryotic structures, and we're going to talk about the organelles that are present within them. So classification, you have prokaryotes. Prokaryotes are part of a domain that they lack a membrane-bound nucleus, and that's important to know. They lack a membrane-bound nucleus. And two of the three domains of life actually are prokaryotes, and those are bacteria and archaea, and that's also important. The difference between prokaryotes and eukaryotes, that's always testable. Prokaryotes are going to lack a membrane-bound nucleus and lack membrane-bound organelles, whereas eukaryotes are going to have a membrane-bound nucleus and membrane-bound organelles. Whenever you think about eukarya, I want you to think about anything that's not bacteria and archaea, i.e. plants, fungi, animals, and protists. So as you can see over here, we kind of have the different domains. We have the eukarya, which is completely separate to itself, and then you have the archaea and the bacteria. And you'll see later on how these two are interrelated. So when we begin talking about cells and prokaryotes and eukaryotes, we need to first talk about cell theory. And cell theory is simple. It states that all things, all basic living units are cells, and that they all come from previously existing cells. And that makes sense when you start thinking about cellular division. Cell theory in itself has been widely accepted for a long time, and it's highly tested. So just knowing that it's a basic living unit, right? We've had this definition previously, and knowing that it comes from previously existing cells. This slide, I just wanted to kind of highlight how small cells can actually be. And as you can see over here, you can see the adult female. With a naked eye, we have essentially around 1 meter. And as you keep getting smaller, 100 millimeters, 10 millimeters, 1 millimeter, 100 micrometers, and so on and so forth, until eventually an atom itself is 0.1 nanometers. And then you can just see the various different sizes. So the slide here has the example of a frog egg being 1 millimeter in size. So roughly around here, the frog egg being 1 millimeter, which you can see it with the human eye, but we can go down even further. And once you get really past this, you're not able to see it as well. With humans, whenever they do in vitro fertilization, when women are having difficulty conceiving, they actually do a procedure where they can extract a human egg and use sperm and try to undergo fertilization. And the egg is so small that the physician actually has to use some type of technology, whether it be special glasses or special equipment in order to see the egg itself. So an electron microscope is typically what is used. You know, the microscopes that we use in the lab, those are not going to be sufficient in order to visualize these structures. Prokaryotic cells. So prokaryotic cells lack a membrane-bound nucleus. Like we previously said, this is going to be one of the most highly tested points between the difference between prokaryotes and eukaryotes. You're going to hear this all throughout if you haven't already heard it in your high school biology courses or previously biology courses. You can see archaea and eubacteria or bacteria are two types of prokaryotes of the three domains. And you can see here there is no membrane-bound nucleus. We have a nucleoid, which is just an area that is not surrounded by a membrane, but it does contain some genetic material. So here we're, this slide is a good slide. There's actually one in your book that's a little bit better, but it shows the different features between, it's a good comparison slide between eukaryotes and prokaryotes. So you have your prokaryotes here, right? Archaea and bacteria both have a cell wall. Bacteria have something called peptidoglycan, and that's important. Peptidoglycan. Um, your penicillins that you use, or you may go to the doctor and take, that actually targets this peptidoglycan, and it breaks this down so that the bacteria essentially succumb to the antibiotic. Um, so it kind of breaks down the cell wall. It's a cell wall inhibitor. Both archaea and the bacteria have a plasma membrane and ribosomes present, and they both lack a nucleus. So, as you can see, the archaea and the bacteria are very similar in structure. The only difference really being the presence of this peptidoglycan. And if any of you guys are going to go on to medical microbiology, you guys will talk about peptidoglycan more. For our purposes, I just simply want you to know what peptidoglycan, just to realize the word, that it's present in bacteria, not in archaea. For eukaryotes, the major thing here again is that the nucleus and the membrane-bound organelles are present. As you can see here with the prokaryotes, we did not have a membrane-bound nucleus or membrane-bound organelles. And of course, the plasma membrane and ribosomes are present as well. We know that the ribosomes are going to be responsible for protein synthesis, as we'll see later on, and the plasma membrane is responsible for permeability. One notable feature that is important, not necessarily test-worthy, but it's nice to note, is that the eukaryotes have a larger ribosomal unit than the prokaryotes do. So prokaryotic adaptations, what is this? Okay, the cell theory says that cells are the basic unit of life, and that cells come from previous existing cells. And that's kind of the what we're going to hit on, is that the development of eukaryotes actually came from prokaryotes. But how? Well, it's through this evolutionary lineage, as we saw in previous chapters, that we had something called the endosymbiotic theory that we'll discuss later on. But essentially, the archaea were able to live in these extreme environments. And this is actually an important point that I want you guys to make note of. This will be on the exam. They were able to survive in extremes of heat and pH and salinity. And it was due to this spanning of the lipids that they had that made them able to survive for such a long time. And here I just have essentially the prokaryotic cell structures. Again, bacteria come in different form. They have something called coccus. You've heard of Staphylococcus. It's what's responsible for MRSA, methicillin-resistant Staphylococcus aureus. Coccus is simply another word for spherical shape. Bacillus is just rod. You guys can look these up. I'm not sure if I included pictures. There's two other types, a spirochete and a spirillum. Not really important for you guys, just wanted to include this. Prokaryotic cell structure. So we have a cell envelope, consists of three different portions. You have the plasma membrane, the cell wall, and if it's present, the glycocalyx. What is important for you guys is I want you to know what the plasma membrane is composed of. It's composed of a phospholipid bilayer. You have these hydrophilic heads, these hydrophobic tails, as you can see in the orientation here. The hydrophilic polar water-loving heads are oriented towards the cytoplasm, which are on the outside portion here. And then you have these hydrophobic tails. This is going to be responsible for the permeability. This will, this polarity within the phospholipids will dictate what is able to enter and what is able to exit the cell. Just to note here, this is a great picture because here you can see different things that we're going to talk about later. You see a glycoprotein, which is just a protein attached to a carbohydrate. That is formed within the Golgi apparatus that we'll discuss later on. The Golgi apparatus is just a assortment center. I want you to think of it being like a factory of the cell. It's responsible for bringing in different proteins, modifying them, and sending them to their destination. This is a glycolipid. This is a lipid with just a carbohydrate attached. Again, the attachment of the carbohydrate takes place within the Golgi apparatus. You can see here we just had the bilayer, bi meaning two layers. Cholesterol. Cholesterol is an important portion that makes up the permeability of the bilayer itself. It's responsible for the permeability. You have integral membrane proteins. These are just proteins that span the entire membrane. And then you see peripheral membrane proteins. These are going to be responsible for binding of ligands, binding of different receptors, be responsible for cell-to-cell interaction, things like that. I'm getting into a lot of nitty-gritty details. I just want you to realize this is a bilayer. This is a phospholipid bilayer. That's important, phospholipid bilayer. Here we have a slide just kind of mentioning about the glycocalyx. It's a layer of polysaccharides that lies on the outside portion of the cell wall. In some bacteria, it's not present in all. When the layer is organized, it's not easily washed off. Sometimes it's called a capsule. Definitions are always good exam questions. I want you to realize what a cytoplasm is. As you can see here, the cytoplasm is just going to be the semifluid type of layer with inorganic and organic molecules. Knowing that it's a cytoplasm, semifluid cytoplasm. As you can previously remember, we said that prokaryotic structures do not contain membrane-bound nucleuses. So what do they have, Shay? They have a area called the nucleoid. And the nucleoid is going to be composed of genetic material as well. This is a DNA region within the cytoplasm. So just knowing that nucleoid, prokaryotic, nucleus, eukaryotic. And then I have down here the mention of something called plasmids. Again, if you're taking microbiology, you'll learn more about that, but I included it in here for completeness. Not going to really test you guys on that. What is important are these two things. These are on the surface of the cells. It's responsible for movement. So when you hear about flagella and fimbriae, I want you to think about movement. Flagellum is this long portion right here. This kind of looks like one of the creatures on All Real Monsters, I think. But you can see right here, this is just a long whip-like structure that acts kind of like a propeller. It's just responsible for movement. And then fimbriae, I don't have a picture of it. So obviously I'm probably like flagella a little bit more, as you can see. Fimbriae are just short appendages that help them attach to the appropriate surfaces. So now that we've covered prokaryotic cells, let's get into the eukaryotic cells. And the eukaryotic cells are what we have. Again, it's the fungi, the animal, and the protist, some plants. But what distinguishes, again, like I've mentioned for the third or fourth time, is the presence of a nucleus and a membrane-bound organelles. And you're asking, what are organelles? Organelles are just these subcellular compartments where these chemical reactions occur or these modifications occur. You've probably heard of things called mitochondria, Golgi apparatus, ribosomes, nucleus, things of that sort. We'll cover these in greater detail. I just want you to familiarize yourself. But on this slide, the presence of a nucleus, the presence of membrane-bound organelles, that's what's important. And again, down here, we have the different groups. I would know these. I think these are important to realize what makes up, what subgroups are consistent with eukaryotic cells. Here, I just have, there are some exceptions, but for our purposes, I just want you to associate that eukaryotic cells typically have a cell wall. Organelles refer to only membrane-bound structures within a cell. It has come to mean any well-defined subcellular structure. The nucleus. Okay, this is an important slide. So what is the nucleus? The nucleus is going to be the defining characteristic of the eukaryotic cell, right? That's really what separates prokaryotic and eukaryotic structures, is the presence of a nucleus. And within the nucleus, you have genetic material. And we know that genetic material, as we previously discussed, is DNA. And do you guys remember what DNA consists of? A phosphate group, nitrogenous base, and a sugar, right? Good. There's something called chromatin. That's important to know. Chromatin is just DNA and some associated proteins. You're going to hear later on about something called euchromatin and heterochromatin. Heterochromatin is inactivated DNA, whereas chromatin is activated DNA, or DNA that is genetically expressed. But within this, DNA will become coiled, and it will be condensed into structures called chromosomes, right? Chromosomes. And we know that chromosomes are important because we have 23 pair, we have 46. Unless you have Down syndrome, then you have an extra one. Then you have 47 chromosomes. And we'll discuss how that happens later on. But for this slide, nucleus is a defining feature of eukaryotic cells. It contains genetic material, something called chromatin. Chromatin consists of DNA and associated proteins. Here I have another slides about what the nucleus is and the nuclear envelope. The nucleus is a region where a special type of RNA is produced called ribosomal RNA. So I want you to think nucleus or RNA. Nucleus, ribosomal RNA. That's where it's produced. That's all I want you to know about it. Nuclear envelope. This is a membrane that separates the nucleus from the cytoplasm. So the nuclear envelope envelopes or closes off the nucleus from the cytoplasm. Just knowing this. Double membrane that separates the nucleus from the cytoplasm. And you can see here the nuclear envelope is right around here, and it's separating this nucleus within here from the cytoplasm, which is present on the outer portion. Ribosomes. Ribosomes are important. What I think you should know about them, they produce proteins, responsible for the synthesis of proteins. There are some different little nuances about them, but for you, they're responsible for the synthesis of proteins. That's all you need to know. This is a complex series of trafficking that happens within your cells. This is kind of a complex series of systems that we have. We have something called the endoplasmic reticulum. You have the Golgi apparatus. The Golgi apparatus always looks like this weird jellyfish-like shaped structure. You have the rough ER and the smooth ER. We'll talk about those, lysosomes and vacuoles. We're not going to really go into detail. I'll just give you some highlights of these, what I think that's important. But this is a communication of, or it is a trafficking series in which proteins are able to move throughout the cell. So in the endoplasmic reticulum, there's two different types, the smooth ER and the rough ER. The rough ER contains studded embedded proteins, whereas the smooth ER does not. The smooth ER is responsible for detoxification, whereas the rough ER is responsible for protein synthesis. It makes sense that the rough ER is responsible for protein synthesis because it has proteins embedded within it. I want you to just realize that the endoplasmic reticulum exists. It looks like a series of flattened sacs. Why is it important? Well, there's something called cystic fibrosis. In cystic fibrosis, there's a mutation in a plasma membrane channel protein. I'm not going to test you guys about this. I just want you to realize its application. But cystic fibrosis is this disease in which there's a abnormal point mutation that occurs. And a point mutation is simply just a mutation that changes the coding for a protein structure. So you get a misfolded protein, and it's usually a chloride channel. And it's due to a mutation in the CFTR gene. It produces this abnormal chloride channel. And within the endoplasmic reticulum, that's where the protein is produced. And whenever this chloride channel doesn't work, they are not able to push chloride to the surface. And you're asking, well, why is that important? Well, chloride is going to be extremely important in mucus production. So if chloride's unable to be present within the mucus, it can't become very lubricated. Therefore, it's thick and sticky. Chloride's also responsible for the secretion of pancreatic enzymes. Pancreatic enzymes are responsible for breaking down food after you ingest it. So therefore, they can get this backup of pancreatic enzymes within their pancreas, and they can get pancreatitis. They can get something called malabsorption, where they don't necessarily digest their food and they're very thin. And you may know somebody with cystic fibrosis. They're prone to pneumonia just due to the fact that they can't clear their airway because of this thick mucus. This is a chest x-ray of somebody with cystic fibrosis, and it's all due to this mutated chloride channel that you see all this scarring of the lung here. Again, not necessarily important for you guys. I just want you to see its clinical application. Here, this is again, the rough ER. The rough ER is studded with ribosomes on the side of the membrane that faces the cytoplasm. I just want you to realize that protein synthesis occurs here. Smooth ER, continuous with the rough ER, but does not have attached ribosomes. That's a major thing. The rough ER has attached ribosomes. The ribosomes are responsible for protein synthesis. The smooth ER does not have ribosomes. It doesn't produce proteins. Instead, it's responsible for detoxification. I want you to realize that, detoxification. The Golgi apparatus. Golgi apparatus, jellyfish-like structure. I want you to think of the shipping center of the cell, because it collects, sorts, packages, and distributes the materials. This is kind of like the post office or FedEx. It's going to get packages in and it's going to send them out. This is something called Tay-Sachs disease. Tay-Sachs disease is due to it's called a lysosomal storage disease. And you're asking me, well, what's a lysosome? Well, you're going to discover that this organelle called a lysosome is responsible for breaking down and digesting proteins. It has these hydrolytic enzymes, that hydrolyze or break down proteins. It's kind of like the garbage dump within the cell. It breaks down lipids, and people with Tay-Sachs disease lack this lysosome, and therefore you get a buildup of this material, and it just deposits in different organelles. And one of the places it likes to deposit is within the eye. Therefore, children will often have eye problems. They have a cherry red spot on the macula right here, and it causes a lot of visual problems, as you can see in these pictures. Usually the infants die by the age of 3 years old, and it's all due to just a disruption in this tiny organelle called the lysosome. That's why it's so important. You see the lysosomes here. We begin discussing about them. Just want you to realize that they contain digestive enzymes that act as a garbage disposal of the cellular factory. A vacuole is just a large membranous sac. Peroxisome, when you think of peroxisome, it ends in zome, sounds like lysosome. Therefore, it contains digestive enzymes. These just break down fatty acids. Also produces hydrogen peroxide.